Hi everyone, my name is Abid. I'm based at Birkbeck College as a postdoctoral researcher in Professor Orlova's lab. And today I will talk to you about the work that I'm doing at Birkbeck, but also highlight the anniversary of Rosalind Franklin. So my journey in science started to pursue a bachelor's degree in biochemistry at the University of Manchester, which then brought me down to London at the ISMB Institute to pursue a PhD in structural biology. I then joined the Orlova's lab to essentially delve a bit deeper into the method, which is electron microscopy. And this method was recognized by the Nobel Committee back in 2017. And it's a powerful method to look at molecules at a microscopic level inside a cell. So before I tell you a bit more about my work, I want to highlight the legacy left behind by Rosalind Franklin's exceptional body of work. Her contributions to molecular biology are far wide, and few of them being her work to determine the structure of the DNA helix, and also coming to join Birkbeck College and leading a group of exceptional scientists to determine structures of viruses, including tobacco mosaic virus. The work that I'm doing right now is looking into the DNA biology and its role in the central dogma of life. DNA is a fundamental molecule in biology which encodes for all living things. And it sits inside a cell in the nucleus on a chromosome. And by having its structure, we know the organization of DNA. So it's made up of two strands which run in opposite directions. And it's made up of four letters, which in combination make different genes. And essentially, the distance between each of those letters on a DNA is around 0.34 nanometers. So here we're talking nanometer scale. And each turn of this helix goes around 3.4 nanometers, and the overall diameter of the DNA is around 2 nanometers. It has two grooves, so the major and the minor groove. And the role of the DNA inside a cell is quite significant. First of all, when a cell needs to divide, it also needs to replicate and make exact copies of the DNA that it has. And this process is known as DNA replication. And one of the important molecules that contributes to this process is DNA helicase, which essentially separates the two strands of DNA. Then we have genes on DNA to be translated into active molecules. And this process is carried out by the molecule called ribosome. And from there on, essentially, events inside a cell which are related to how genes and DNA are regulated and maintained. For my work, I'm focusing on step number one right now, which is looking at DNA replication. And in particular, I'm looking at how DNA helicases unwind DNA in order to start the replication process. To do that, I'm using the method called cryo-electron microscopy. And essentially what we do is we take a sample of helicase molecules, for example, we isolate them from cells and we freeze them in a process known as vitrification. And here we're freezing the molecules in very cold temperatures. And what we then do is we take those samples to an electron microscope and take lots and lots of pictures. And essentially what these snapshots give us is the helicase molecules in different poses and different orientations. And by having those snapshots, we then use a series of algorithms and softwares to analyze those images and ultimately determine structures of the molecule that we're looking at. So here shown is one of the structures of DNA helicase. And so in summary, what we can relate to is the molecule in the context of biology. So in my case, I'm looking at human papillomavirus and the helicase that it encodes. So papillomavirus are precursors of cancer. So by determining the structure of DNA helicases, I'm able to understand how this viral helicase functions and how it allows the virus to propagate. What it will help me to do is to determine therapies and possibly drug molecules that we can design to stop this process and prevent papillomavirus from dividing. So I hope this has been informative for you, and I wish you all the best.